మా ఆఫీస్లో సో ఇక దీన్ని ఓకే కనిపిస్తుంది ఇప్పుడు.
Good morning, dear participants. I'm very happy to introduce my classmate at Andhra University, Dr. Potluri Prashant. After completing his master's degree from Andhra University, he moved to Jawaharlal Nehru University, New Delhi, where he got his MPhil degree and PhD degree. From there, he moved to University of California, San Diego, and he worked as a postdoctoral fellow. From there, he moved to the University of California, Irvine. Presently, he is working as the staff scientist at Center for Mitochondrial and Epigenomic Medicine, Children's Hospital, Philadelphia. So with this brief introduction, I request my good friend Dr. Prashant to deliver his talk on mitochondrial research. So Prashant, the floor is open for you. You can start your talk.
Okay, you can see my slide, right? Hello?
Yeah.
Okay, so, thank you, Srinivas, and indeed it is an honor to present my work here, and I've been working with mitochondria for my almost entire adult PhD, post-PhD work in this country. And today's talk is, just for the general audience. I made it much, more simpler. I am not throwing up all the, crazy techniques we use and everything we did in last 23 years. So as the title says, how do we study and why do we study this mitochondria? Today's outlines of my talk is just to give a brief introduction of maybe 5-10 slides about mitochondria and mitochondrial diseases, and then what is our research, my research and my group's research has done in understanding the biology of mitochondria. And then the third aspect is how we are developing new tools to treat, mitochondrial diseases. In the end, people are, spending lots of money on this research. It's mostly publicly funded research by United States government and Children's Hospital of Philadelphia. And they want us to, advance the field and to bring some, therapeutic strategies so that we can treat patients and public with the advancements we are making. So next slide is basic biology, bioenergy flow in the biosphere. So perhaps all of you are familiar with this slide. Basically, chloroplast of plants, they fix the CO2 with the help of solar energy and generate oxygen and produce glucose as a byproduct of this CO2 they fix. And animals that include us.
Prashant, can you speak a little bit? Can you speak a little bit louder?
Yeah, I can.
Okay.
Yeah.
Okay.
So yeah, and as this slide shows that chloroplasts use the solar energy to fix CO2 into glucose, And the glucose, which is converted into starch and all, we animals and other organisms, we consume this. And in that process, we generate ATP and heat. Both are important for our survival on this planet. So, and the important thing in this slide is both these energy converting organelles have their own DNA and protein synthesis apparatus in them. That is a critical feature that's going to, help us understanding research work and what are we going to use and understand this DNA and protein synthesis apparatus in creating new devising, new therapeutics. Then, as my title said, why do we understand, why do we study mitochondrion? Because in the recent past, the last 50, 60 years, people slowly started understanding that life is a synthesis of information, that is your DNA, and the structure is basically anatomy, both cellular anatomy and physical anatomy, and energy. Often we miss the energy component, and we mostly teach our students and we learn ourselves the importance of DNA or say structure. So if there is a problem with DNA, how do you fix that? Okay, that's well and good, but in last 20-25 years, where you have more tools to understanding the DNA structure and DNA sequencing, people started realizing, people like us, working in this field, started realizing unless you understand the energy component, the vital force that is, putting together all these components and running the cellular pathways, we don't understand the exact science, biology of various diseases. And not only diseases, even to understand basic biology. For example, without understanding chloroplast and the solar energy conversion, plant biology is pretty much, incomplete. So, as I mentioned here, animal cell is essentially two organisms that have been coexisting for last 2 billion years. So, yeah.
Okay.
Okay. Can you hear me?
ఇది కూడా యాడ్ చేస్తారు దా ఫ్లైర్ ప్లస్ ఫోటోస్ కూడా.
Can you hear me? Hello?
Yeah, Prasant. Carry on.
Okay. Yeah. ఏదైనా క్వశ్చన్ ఉన్నా పర్వాలేదు, తెలుగులో అడగమని చెప్పు ఎవరైనా, ఇబ్బంది ఏం లేదు. ఓకే. So, to continue, the symbiotic relationship between an archaebacterium and eubacterium started 2 billion years ago, and that evolved into an eukaryotic cell. In this, the result of this symbiosis led to specialization of nucleus and cytosol from the archaebacterium, which is specialized in the information structure, that is DNA and various cytoskeletal structures. And mitochondrion, which has derived from eubacterium, is totally specialized in converting the energy, basically. And in this process, each cell, the total cell, has close more than 100. It depends on the cell and the tissue. Sometimes, the least is a 100 mitochondria, but the maximum can be, 100,000, close to 1 lakh mitochondria per cell. And you need so many cells to support about 20-30,000 nuclear genes and their products and creating the larger structure for the biological function of the structure. And in the reverse side, while mitochondria, has specialized into energy generation, but it also it derives several, almost 80% of its components from the nuclear encoded genes. As this slide is showing here, this is nucleus, all your chromosomes and all. It produces, RNA and ribosomes. They generate several proteins which go into mitochondria, and mitochondria generates about 13 polypeptides in human and mammalian systems, and several rRNAs, that is 12S rRNA, 16S RNA, and 22 tRNAs. All these components, along with the help from the proteins that are imported into mitochondria, generates a functional mitochondria, which is useful for its function. Then, as this slide shows, the protein synthesis that is being done in the mitochondrial matrix is depending on 200 to 300 nuclear encoded products, and again that comes to about 20% of to this day, say 1200 total nuclear gene products that are needed for the mitochondrial functioning. So, and currently active research is going on in understanding this mitochondrial nuclear crosstalk, or what we call signal transduction. How does the nucleus knows what proteins are required for the mitochondrial function, and how does mitochondria knows how much energy it needs to produce for the survival, for the proper functioning of its host cell. So this is a very active research going on in this country, and several Indian labs are also working in this field at University of Hyderabad and several labs in Delhi. And the next slide explains basically in animals, mitochondria produces 90% of cellular energy. And apart from this energy by itself, the mitochondria are also needed for several cellular processes. For example, apoptosis, that is a programmed cell death. This is a very critical step. Without this, there is no proper control when to stop the cell growth. If the cell is losing control on its multiplication in a eukaryotic system, multicellular system, you don't want the cells to divide uncontrollably. That's cancer. How do you control? And in the recent last 20 years, the research has shown several of these apoptotic signaling molecules are associated with mitochondria, both mitochondrial DNA and several protein components of mitochondria. They act as signals. When these things are released into cytosol, cell knows the pathway, the cell has reached an end point and it should die. So that is very strictly controlled. And in neurosciences and cancer biology, this pathway is extensively studied as a mitochondrial contribution to both these areas of studies. Then, as I mentioned, we all know mitochondria are the energy source for the cell, and through oxidative phosphorylation, it generates ATP synthesis. I'm going to show a cartoon how it works, and a byproduct of this oxidative phosphorylation is what we call reactive oxygen species. And this reactive oxygen species plays a major role during infection, cellular infection and inflammation. For example, the present day pandemic of COVID and all, the recent work, I'm not saying that's not my work in the lab, but one of my colleagues have been actively working in that area. If cells are infected with this COVID virus, the mitochondria generate ROS, reactive oxygen species, as a signaling molecule that is going to, start the inflammatory responses, so that the cells can decide whether to, shut down the viral production, or the cell which is infected need to die, so that the infection doesn't spread to the other molecules. And this is a very active research, and the papers are ready to be published. And our lab has clearly shown if you can modulate mitochondrially generated reactive oxygen species, you can control the viral proliferation and perhaps the disease progression too. And another important disease our lab and several other labs in this country have been involved is the Parkinson's disease. It's a neurological disease where you have uncontrolled muscle shakes, And to this day, people are struggling to understand what is the root cause of this disease. And there are all types of theories and all, but the only aspect that could be reproducibly, worked on that can generate and treat Parkinson's is mitochondrial loss. If you have, say, mitochondrial defect that is generating lots of mitochondrial loss, that can generate Parkinson's. And we are not, our lab is not currently working on that, but our model systems we generated in the lab has clearly shown that is indeed a case in several. Parkinson's is a complicated disease and it has several aspects, and there are several varieties, but a major chunk of Parkinson's can be explained as a mitochondrially generated ROS that is starting this disease. And as I mentioned in the previous slides, mitochondrial DNA encodes only 37 genes, but a unique feature of mitochondrial DNA, as I mentioned again previously, it has several copies. Each cell will have, say, at least 100 copies, each 100 mitochondria, and each mitochondria has, again, a dozen to up to 1,000 mitochondrial DNA copies. And mitochondrial DNA in humans and mammalian systems is a circular DNA. Apparently in plants and several yeasts, it could be a linear DNA, but in mammalian systems and humans, it is a circular DNA with 16.5 kilobase pairs, and it encodes 13 peptides and several tRNAs and 2 rRNAs, which are needed for mitochondrial ribosomes. And because these multiple copies of mitochondria in a cell, it is a nice tool frequently used by forensic scientists to identify, the DNA or the identity of the person whose DNA they are testing in case of, parental disputes, or they found a body, or in case of, Egyptian mummies, they want to test, who are the people that have been mummified, and those things have been rotting for last 5,000 years, maybe 3,000 years, and you can still extract a little bit of DNA, nuclear DNA, because you have only 2 copies, and that can be washed away. But with a small material, you can start and look for mitochondrial DNA. You can amplify, and studying closely into the polymorphisms in this DNA, you can vaguely come to a conclusion that these are the people, they came from, say, Africa, or they came from Mediterranean region of either Asia or the Southern Europe. And another important aspect, because it is DNA, this is the rule of thumb in biology. You have DNA means DNA can mutate, and some of these mutations can be bad and they can cause diseases. And that is the basic foundation of this entire field of mitochondrial diseases. We used that hypothesis to check my boss, whose name I'm going to show you in a slide down the road. But he hypothesized in his initial days that mitochondrial diseases may cause human disease. Mitochondrial DNA may be a cause of certain human diseases. And this slide shows mutations in mitochondrial DNA. Again, mitochondrial. The root of that hypothesis is initial. The early scientists in this field, they realized mitochondrial DNA doesn't have a proper DNA repair mechanism, which is existing in cytoplasm and the nucleus that can repair any damage to the nuclear DNA, but you don't have that system in mitochondrial DNA. So because of this lack of DNA repair mechanism, mitochondrial DNA accumulates at much higher rates, this mutation load. And as I said, some of these mutations are often pathogenic. So because, almost all these genes that are present in human mitochondrial DNA are critical in energy biogenesis. And any damage to these genes, say to one copy or two copies, or say 30% of these DNA copies, if they are mutated, they could cause a disease. I'm going to show you some data out of that. And and another important aspect is, as we age, mitochondrial mutations accumulate. That's the general property, because it's a highly oxidizing environment we live in, and 21% of oxygen and various UV in the atmosphere, or the chemicals in the food or in the air, they can cause mutations in any DNA, and more so in mitochondrial DNA. So as we age, these mitochondrial DNA mutations accumulate, and we have shown in our lab with experimental proof, both in mouse models and human patient studies, that these age-related mitochondrial DNA mutations can cause diseases. And in certain mouse models, if we interfere in this DNA mutations, stop or prevent or treat these mitochondrial DNA mutations, you can treat some of these aging related mitochondrial degenerative diseases. And the map here shows, as I mentioned, these newly created mitochondrial DNA mutations are highly adaptive. What does it mean? Because as the modern anthropology shows, all the human origins are in Northeast Africa. And all of our ancestors, both whoever is living on this planet, the humans, they all originated in that small corner of the globe in Africa. And they went and settled in various climates, all the way from Siberia, cold, to hotter tropical countries like India, Africa, it's a tropical continent, and to Siberia, to high altitudes like in Tibet, and down in South America, the Andes and all. So the current hypothesis is some of these mutations are highly adaptive. For example, some mutations we recently studied that affects complex 4, which is sensitive to cyanide toxicity, has adapted to high altitude survival in Tibetan populations. Also, others have noticed that when you are living in colder climates, the mitochondria has adapted to that colder climate by generating heat instead of generating ATP. So they can eat lots of fatty food, lots of sugars, but instead of gaining weight, they can use that excess calories to generate heat, and that is highly beneficial for survival in those hostile conditions. And this is a controversial, not controversial, it is accepted, but to generate that type of proof is little tough. But what we observe in various geographical association of mitochondrial DNA polymorphisms is a well-established science, and because of that, you have certain mutations associated with these populations that leads to various diseases. And outside this ring is noted as various nucleotide positions and the mutations that have been extensively studied by various medical groups, which causes human diseases. For example, LHON, this is Leber's hereditary. That means you get it from either your mom or dad. Optical nerve damage, and that is caused by mutations in this nucleotide position 14,484. And if you have that mutation, and if you are male, once you are past 30, suddenly you get up one day, you cannot see anything in one eye. Within a couple of months, the second eye also goes blind. And until recently, people could not recognize what is the problem, because they could check all the known genes in nuclear DNA. They could not find any problem, any of the DNA. Then my lab and labs in UK and all, they have shown this is exclusively mitochondrial inherited disease. And specifically this gene called ND6 that makes this enzyme NADH dehydrogenase. If you have any problem in this gene at this position, that is going to cause an optical nerve damage and you go blind. Similarly, there is another disease called this deafness and dystonia that is caused by a mutation at this position. And this is even surprising mutation. So if you have this, you have no problem with your hearing and all, but your mitochondrial DNA mutation at that position makes you sensitive to several regularly used antibiotics. So unknowingly, people use some antibiotics like penicillin and all, what we call aminoglycosides. These things, when you use that, suddenly you go deaf. So, and people struggle for almost like 30 years. What is the connection? Why do you go deaf only after you use these antibiotics? Now we know, because there is an underlying mutation that makes you and your mitochondria sensitive to that antibiotic. But because, as I said, these mitochondria are derived from the bacterial ancestors, and say to this day they retain the same sensitivity to antibiotics. And in humans, when they have this mutation, they go deaf. So these are all fascinating areas of study. Now people are slowly understanding, and if they find somebody with that mutation through genetic screening and all, they advise them, don't use these certain antibiotics. Or if you use, you go and run to a doctor so that they can prescribe some medicine, so that you don't go totally deaf, or your children can survive. That's one aspect of this interesting science. And this is more again related how you get mitochondria from your parents. As we understand now, most of the higher mammals, that including humans, you get your mitochondria exclusively from your mom. Dads have no role in this mitochondrial DNA inheritance. The dad's mitochondria is stopped outside the egg, and only nuclear DNA goes in and fertilizes. And the mitochondrial DNA that is exclusively inherited from your mom gets into your body. And if you are a female, you will give it to your male and female children. And if you are male, okay, that's the end of your mitochondrial DNA study. So this map shows, this is the original contribution of my supervisor, my boss, Professor Doug Wallace. He is the first scientist to recognize that humans inherit mitochondria exclusively from their mom. This pedigree map shows it. So you can see in the red circle, the mitochondrial DNA inherits only through the mom to her kids. And when their kids, they get married, if it is a female, a daughter, she inherits those DNA and it goes to her children. And male, it's a dead end. You don't, give those mitochondrial DNA to your son or daughter. And another aspect of interesting aspect is, you, as I said, you get thousands of copies of this mitochondrial DNA. And say your mom has mitochondrial DNA mutations. How does your system screen for that type of mutation load? We don't know exactly how the body screens for that, but it seems there is a cellular system that screens certain percentage of mitochondrial DNA mutation load that may be interfering with, say, oxidative phosphorylation, or maybe it is generating lots of reactive oxygen species. And cellular systems recognize that and destroys that mitochondria and the cell that is containing so much load of mitochondrial DNA. And this is a fascinating science. And when initially these things were published, people were not ready to accept it. But slowly, with the modern DNA sequencing techniques and all, they observed the same phenomena in aging bodies and all. So now we know this is indeed a very valid aspect of mitochondrial DNA. And mitochondrial DNA derived mitochondrial diseases, both as aging and inherited mitochondrial DNA diseases. And another, as I mentioned, convenient tool is because you inherit your mitochondrial DNA exclusively from your mom, it's very easy to build your pedigrees. So, and that's how they devise various populations, where they came from and whose their ancestors came from. And all these things support the now accepted scientific hypothesis that all humans have, migrated, moved out of Africa. So this is a very fascinating study. We used to work on that, but now we are working in a hospital, so they want us to, work on human diseases and derive some therapies for that, for all the money they are spending on our research. Again, an important point here: mitochondrial deficiencies, or what it could be mostly energy deficiencies, impact mostly energy demanding tissues. Most of your energy demanding tissues, muscle, brain, you need lots of energy to think and to digest all the information you get. Your digestive system to take all these nutrients and all. You need lots of energy, both to make the various enzymes that are required, and heart, it needs to function from the day one until the end point, and your eye, because what you are seeing, all your optic nerves and all, they need to convert that data into a visual image that can be sent to the brain, so that if you see a tiger, you can run away from it, or if you see a snake, you can be scared not to touch it. So, but most of these typical mitochondrial energy deficient diseases, they start with this energy demanding tissues first, and when we do our research, we say when we create a mouse model, the first thing we test is if there is an obvious observable cardiac phenotype, heart phenotype, or if the mice has some various crazy digestive problems. The mice, even if it is a small animal, you can, when you observe closely, you can really see there is some problem for these mice. And we generated various mouse models to test various aspects of progression of these diseases. I'm going to show you a couple of models. And based on that, our thing, in fact, in our lab is considered one of the pioneers of generating these interesting models, which can emulate or reproduce some of the human patient data, so that we can move back and forth. So if we notice something, if we devise a therapeutic strategy, say chemical therapy, we can test that first in the mouse models, and if it is looking promising, then we can move on to human studies and all. Then okay, it's all well and good, we know the mitochondria are important, but what are the techniques we use? Currently, the core of mitochondrial research is biochemistry and molecular biology. We are using next generation DNA sequencing. We have a very good facility in our lab, in our center, but before we go there, first is just to take some tissue, say from a patient. We can easily take a small skin biopsy, and then we have very sensitive machinery in the lab. We can test those cells in whether the mitochondria in this human sample is respiring properly. If we notice that there is some deficiency, which has been studied extensively by other groups and all, then we will go ahead and use various molecular biology techniques like DNA cloning and all, and sequence that specific gene or known genes, unknown genes. And by putting together all this, all this information, we go to the understanding. As you can see, you're all familiar with this famous scientist Pallad in cell biology studies. He is the first gentleman who could observe under electron microscope. There is a mitochondria, it has cristae and all. And the further improvements in the new computer-derived imaging and all gave us much better information about the structure of this cristae. and various sub-compartments within the cristae and all. These are immensely helpful in understanding various deficiencies or the structural details or functional details of mitochondria. I have always used various aspects of oxidative phosphorylated for electron transport chain, and all inside the inner membrane of mitochondria. As this slide shows, you have an outer membrane and you have an inner membrane, and from the inner membrane you have the cristae that goes into the matrix, and these cristae membranes, they host most of your oxidative phosphorylation enzyme complexes. And some of the data I'm going to show in the next few slides, they're mostly about the complex 1, which is an NADH dehydrogenase. NADH and all other reactive molecules, they are derived either from Krebs cycle or glycolysis or fatty acid metabolism. They're pumped into the electron transport chain, entering through complex 1, complex 2. Complex 2 is a succinate dehydrogenase. Complex 3 is a coenzyme Q cytochrome C oxidative reductase. So it's going to accept the electrons from complex 1 and 2 and then transfers them to complex 4. Complex 4 takes the electrons from cytochrome C and uses oxygen as an electron acceptor and generates protons. And the protons derived from complex 1, 3, and 4 are used by complex 5 to generate ATP. And ATP, we all know as biologists, that is the currency of your cellular energy. And we studied oxidative phosphorylation as a core technique, because this technique has been developed between mitochondrial and chloroplast biologists for the last 100 years. The instrumentation is very cheap and the chemicals are easily available. So if, say, we receive a patient sample of unknown mitochondrial deficiency, the first thing we test is see if complex 1 or complex 2 or complex 3, any of these complexes is not functioning properly. We can easily assay for that, and because of the ease of assay here, we can move on. If everything looks normal, then we can go to the more sophisticated techniques for which we have more instruments, and they are expensive. But once, most of the time you can catch there is some deficiency in and around these complexes. So, and complex of my interest when I joined the mitochondrial biology research is complex 1. And complex 1 is NADH dehydrogenase. And why is it important? Because at that time, even to this day, majority of frequently diagnosed mitochondrial diseases are due to a complex 1 defect. And complex 1 is, as I said, is an NADH dehydrogenase. It has 45 protein subunits, and it needs close to another 50, 60 protein cofactors as assembly factors that need for its proper assembly. And my work and my colleagues' work has clearly shown 5 or 6 of these peptides. If you have, say, some certain mutations in these peptides, you have a severe mitochondrial disease. And I'm going to show you some data we generate based on this information we derived from the patient studies. We made mouse models and we recapitulated most of the patient phenotypes. And another important aspect of this complex 1 is 7 of its 45 subunits are made by the genes located in mitochondrial DNA. That's an important factor because I'm going to show you some slides down here. So while studying this complex 1, I got across an interesting patient cell line in which, and we have thousands of isolated mitochondrial deficient patient cell lines. I started screening them using various protein and DNA techniques. we basically made them into three groups. One is where you have only nuclear DNA is carrying the mutation. Various, as I said, each of these complex one genes, you have 45 of them, 38 of them are from the nuclear DNA. So this cohort of patient samples, they have clearly nuclear DNA deficiency. And the other extreme is where you have a mitochondrial mutation, and that is causing a mitochondrial disease. Then there is this interesting patient sample where we have partial mutations. Partial means the mutation by itself is not very lethal, because we have experience with working with those type of mutations. And at the same time, it has another nuclear mutation by itself that is again not very severe. But the patient is showing all the severe phenotypes of a mitochondrial disease. So we hypothesize maybe these partial mutations coming together is generating that severe phenotype. And the nuclear mutation we identified in this poor patient is one is a complex one peptide called MWFE, and we observed that there is a very evolutionarily highly conserved glycine at the position 32. It is mutated into arginine in this patient, and because by then we already have a mouse cell line which has the same mutation, and that cell line has shown, it's a rodent cell line, shown that it has a partial deficiency. These are all various protein chemical techniques we are using. This is a classical SDS Western blot, and this is what we call native gel, in which you can separate these various enzymes in their native functional stage, and you can separate them on a gel, polyacrylamide gel, and then you can assay for each of these enzymes in the gel, and you can visualize whether the enzyme of your interest is functioning or not. So as you can see in this mutation, where what we observed in the patient, the complex one, that is Nadh Aerogenesis, is severely downregulated, means it has gone down, and that is reflected in when we measure again the respiration in an oxygen electrode. If you have this mutation either in human cells or in mouse rodent cells, the complex one respiration has gone down substantially, and that also impacted the cells growth in a petri plate. You can take these cells and grow them and see which one is growing faster. So if you have a normal wild type MWFE, this is a peptide, your cells are growing at certain rate, and if you have a mutation in this peptide, that means this is, but as I said, in this patient the deficiency is much more. So we have another technique that's being both in our lab and in our collaborators lab, where you can move the mitochondria from one cell to a different cell. You can fuse the cells in a petri plate with a donor cell where you don't have a nucleus. You can use certain chemicals that can destroy the nucleus in those cells, but it doesn't touch any mitochondria. And you can create a similar sister cell line where you can destroy mitochondria and mitochondrial DNA, but it doesn't damage the nucleus. Then we can fuse those things, those two cell lines, where your only variable is your mitochondria that carries these mutations. And if the patient so we unlucky and you get your mitochondrial DNA from your mom, and you get your and that also impacts the same enzyme, you mitochondrial disease. And in this several metabolic disorders like diabetes, and he has a moving disorder, so he cannot walk without support, and these are all typical classical mitochondrial deficiency disorders. We thought the important observation we made was all his maternal cousins, not all, several of his maternal cousins and maternal uncles have the same problem. Maternal uncles were also mentally retarded. They died rather early in their life, say 40, 45 years, and they have same mental retardation. So that clearly shows the deficiency which we observed in the mutations which we observed in the mom's mitochondrial DNA is causing the disease. So based on this information, we went ahead and created various mouse models. We created one mouse model which has the same mutation we observed in the previously shown the patient work, and that also generated a similar phenotype. The mice have, when you observe it, they don't have any problem per se, but and behavioral changes. They have been feeding off all over the place. Typically, mice, they feed during the night, and apparently these mice, they feed all at whatever time they like. And that is a observation dot. So that means we could recapitulate the same. And another interesting aspect is certain mitochondrial DNA mutations, they do not impact any known functions. We know these are mutations because that is not existing in the wild type population, and it is not affecting any mitochondrial respiration or other known enzymatic functions. But when we measure the reactive oxygen species that is generated in these mitochondria, they are alterations. And this ROS phenotype to show you in the next slide. So, for example, this lawn phenotype. As I said, this causes blindness. But surprisingly, when we made the same mutant mice with the same mutation in mitochondrial DNA, it has no complex 1 deficiency. Again, as I said, this is a complex 1 specific mutation. There's no complex 1 deficiency, but we notice there is a huge ROS production in these mice, and we hypothesize perhaps this ROS is damaging the optic nerve, and which is leading to the blindness in the human patient. In fact, when we extract the eyes from these mouse models and send them for the electron microscopy and various other sophisticated studies, we noticed a severe damage to the optic nerve. But as I said, complex 1 for its function like Nadh Aerogenesis and respiration is not severely altered. So that means we hypothesize perhaps this mitochondrial DNA damage or mutation is generating lots of ROS, and the ROS is damaging down the road in conjunction with some other nuclear or environmental factors, damaging the optic nerve and other diseases. So now I'm going to the final part of our research work. So okay, we gained all this knowledge. What do we do with all this information we gained from either patient studies or the mouse models or the patient cellular studies? And we have several other models. We can study some of these diseases in flies too. Drosophila, we can because they conserve some of the same proteins and which work the same, which do the same function in mitochondria. So, and with all these models, we gain some knowledge. How do we treat? That's what the hospital wants us to devise some strategies, because some of these mouse models are very well regarded These were made almost 30 years ago, and we share these models with labs all over the globe. In fact, some of the Indian labs, they borrowed these mouse models we generated in the lab, and they're using for their studies in their lab and various other aspects. And in our lab, we are using these models to develop new treatments, how to treat in either patients and all, because we cannot directly go and treat with unknown therapies into the human patients. So we are using the lab-generated mouse models and to test various gene therapy strategies. We can use chemicals too, but pharmacological impact on mitochondria, it is all over the place, because we're not sure there are off-target effect on all these chemicals. So we don't want to create a secondary problem with these chemicals. So we thought we should go for the gene therapy. What is gene therapy? Gene therapy is nothing but if you find a mutation in a patient or in a mouse model which is causing some deficiency, and you can substitute that gene with a wild type copy of the missing or defective gene. This is a science which has been in various forms that is being employed for last 30 years, but in last 5-10 years they are very well established, and FDA, that means the American Food and Drug Administration, accepted this as a therapeutic. That means they have done extensive studies on human patients. So as a treatment therapy, this is safe. So we are adapting this strategy to develop new therapeutics. As I said, we have several mouse models. At this time, we have 6 unique mouse models, and total 10 mouse models with of various mitochondrial DNA, nuclear DNA mutations, and various forms of mitochondrial diseases. And of them, in our lab is, say, if you have a nuclear gene mutation. For example, in one of the models, which is now considered as the best model anywhere in the world for the mitochondrial diseases, called ANT1. That is an ATP-ADP translocator that moves ATP-ADP in and out of mitochondria. If you have any mutations, it causes a heart enlargement and other mitochondrial, typical mitochondrial disease symptoms. And we generate a mouse if that is a nuclear DNA mutation. So that can be easily treated with these new gene therapy tools that has been developed by other labs. The unique thing about mitochondrial DNA mutations is mitochondrial DNA, you cannot repair or substitute the DNA deficiency in mitochondria, because mitochondria does not allow external DNA. You can push mitochondrial DNA into not be functional inside the mitochondria, the foreign DNA. And for that, we are devising a novel strategy where we are taking the mitochondrial DNA derived protein and engineer it so that that can be made in the nucleus. And then if you have a mitochondrial targeting peptide sequence, these are well studied amino acid sequences. If you like it, if you add these unique sequences to your protein of interest, that protein can be transported into mitochondria. So we are trying to see if the mitochondrial DNA deficiency can be repaired by supplying that missing protein through the nucleus, through the cytoplasm, into the mitochondria. That is still at the very preliminary stages, because mitochondria, it looks like, does not like even the protein beyond a certain point. You can, for example, send some unique enzyme, it goes in and does it work? But it's not so that even other that can be used as a therapeutic for mitochondrial DNA deficiencies. And then recently we stumbled on RNA as a molecule, because mitochondria, both in human and yeast, are known to take up RNA much more easily than DNA. So we thought if we can engineer RNA with several modifications, and we can push it into mitochondria, and mitochondria, as I mentioned, has its own ribosomes and all. Maybe mitochondrial ribosomes can make proteins out of this messenger RNA that we are pushing from outside. This is also a new strategy. We have preliminary data, they're telling us that this is doable, but we have not reached a point where we want to go to the mouse models yet. We are still, optimizing these techniques in the cell culture models. So again, as I said, this is gene therapy that is, universally used. Basically, they used this virus called AAV, adeno-associated virus number 2, and we take that virus and remove some of the original genes from this virus backbone and put your gene of interest, our gene of interest, and then make viruses carrying our gene of interest, then inject into mouse or the human patient tissue and see if the gene can substitute the mutant deficient gene function and repair the mitochondrial damage. So as I said, we are using one of the lab generated mouse model that has to do with ANT1. This is a ATP transporter, and parallelly we started another strategy. As I mentioned, in certain mutations, reactive oxygen species is a culprit, which is starting the damage. So we hypothesized maybe if we can find a way to stop that reactive oxygen species in mitochondria, maybe we can, stop the damage to the optic nerves and all. So we are engineering new enzymes that can selectively repair the oxidative damage caused by the reactive oxygen species. These two are the most advanced stage of work as of now. And as I said, ATP ADP translocator is on the mitochondrial outer membrane, and it's a nuclear gene. And again, it impacts mostly muscle, that includes heart. And it's a well-known human mutation that causes cardiomyopathy, that is heart enlargement. And in these patients, in certain subset of patients, if they don't receive a heart replacement, means in a donor heart, they will die by 30-35 years of age. So so the heart enlarge grows big. I'm going to show you the next slide. Grows so big it is not functional at all. So we generated a same mutation in a mouse model, and it has the same heart enlargement problem, and it also dies within how normal average mouse can survive for say 2 years. These mice can barely survive for 1 year. And along with the ANT1 nuclear mutation, if you have say another mitochondrial DNA mutation, this can barely survive beyond 3-4 months. So that means these mutations are very severe, and they are one causing the disease. So if we can somehow repair this DNA mutation, can we stop the disease progression? So with the help of my colleagues in the lab, this is the mice heart. These are the exact size of each of these heart. This is the wild type heart of a 6-month-old normal mice, and this is the ANT1 deficient mouse heart. You can see it is severely enlarged. And if you add to the ANT1 mutation in mitochondrial complex 1 specific mutation, it is going like balloon. So it is a very severe mutation. We could, easily observe this phenotype. So one of my colleagues, she is a specialist in handling these mice, because mice is big, 3-4 inches size, and mice pup is even smaller, maybe 2 centimeters. And you can imagine injecting some virus into the heart of that pup, which is barely 1 or 2 centimeters. And I have a colleague who mastered this art of injecting a 2-3-day-old mouse pup's heart, the virus carrying ANT1 wild type gene. So again, she went and injected this wild type copy of ANT1 gene, and it looks like, especially this ANT1 ND6 mutant mice, you can almost 100% reverse heart damage. And the same thing is observed in the heart function. This is the EKG, echocardiogram, and you can see this is a wild type heart. This is the ANT1 ND6 mouse heart, which is ballooned. And when you inject this heart, this mice with the virus carrying the therapeutic gene, the heart size is reduced. It's not going back to the normal, because the damage is already done, but it seems it is bringing back some of the normal size of the heart. Again, my colleague Alessia, she measured the heart function, that ejection fraction, how much blood it can pump, and cardiac output, various amounts at different stages of post injection. So you can clearly see when you inject the virus into the heart of these various mutants, this red and green, the function is getting back to more or less normal levels. So that means this strategy of devising, this is a very expensive proposition, because, for example, all this part of the study, we spent $4 or $5 million to make the mouse model and to get all these gene therapy vectors developed, and the manpower, reagents, and all. And when we go to the human patient studies, we may end up spending another $10 million. But they won't allow us to go to directly the patient studies unless we show reproducibly that in a mouse model carrying the same mutation as the patient, we are, treating the, a genetic defect caused by this mutation. So we are very excited. This is a new data. We have not published this data yet. And the hospital, which pays most of our research funding, is very excited, and they want to support us for a few more years so that we can take it to the patient treatment level. And we are very excited. And we have the other that is, we are trying to treat the optic nerve damage in the mouse. That is at the preliminary stage. Because, again, as I said, to make these mice and then treat them, inject each study. If we need to study, we need to treat at least 30 to 40 mice for each of these age group. So, and then we need to have people who can study all these things and then get this data. This slide, it took us nearly 4 years to reach to this point, from developing the viral vectors and breed the mice to that level, then we inject and wait for 9 months, then send, develop various techniques to study the restoration of this function. So in conclusion, this field of mitochondrial medicine is filled with challenging questions. But if we can find answers, even not for all the diseases of mitochondrial origin, but even if we find some therapeutics, they have the peripheral benefits of, as I said, in aging, mitochondrial deficiency is a big factor. Maybe down the road, maybe in 20, 25 years, these therapeutics which we are developing in the lab can be used to treat aging associated mitochondrial diseases too. So I want to thanks to my boss. This is Professor Doug Wallace. He's a pioneer who single-handedly created this field of human mitochondrial diseases. And with his vision and guidance, we could establish a very successful center, and on which we spent close to $100 million in last 10, 12 years. Nadh is my old colleague from India. We did PhD together, and he also studies mitochondrial diseases. He was very instrumental in developing some of the mouse models. And Dr. Alice Angel, she's an Italian scientist. She's a specialist in, my specialty is generating all these viral gene therapy vectors and studying the protein chemistry and biochemistry. She is specialized in injecting into the mouse heart and then, go and assay various aspects before killing the mouse. And she's and I want generous time and the opportunity. Thank you.
So thank you, Prashant. Now the lecture is open for discussion. Dear participants, if you have any queries, you can pose to my friend Dr. Prashant.
తెలుగులోనే అడగండి, పర్వాలేదు.
So if you don't have any questions, I will conclude the talk.
Okay, thank you, Srinivas, and thank you for the opportunity.
Just a minute.
Sure, please.
So now, Dr. Prashant, starting from how the mitochondria entered into the archaebacteria and it became endosymbiotic, and how it is producing energy, and he also explained how it is important in suppressing certain diseases and also responsible for causing certain diseases. So this is a highly informative talk on the mitochondria. So I am very grateful to my good friend Dr. Prashant for delivering a nice talk, coming down from his scientist level to a student level. So thank you once again, Prashant.
Thank you all, and thank you, Srinivas. And it's a very exciting opportunity. Thank you.
Thank you. It's a privilege to us.
Okay, thank you. Bye.
Okay. Thank you.